In transcription, the DNA double helix opens along its length. One strand of the open helix remains inactive, while the other strand acts as a template against which a complementary strand of RNA forms. Transcription is performed by a special enzyme called RNA polymerase. The RNA polymerase attaches itself to a specific DNA nucleotide sequence situated just before the beginning of a gene. This specific sequence is called a promoter sequence. The RNA polymerase starts constructing RNA using ribonucleotides moving freely in the cell. The sequence of bases along the RNA strand is identical to the sequence of bases along the inactive DNA strand, except uracil, or U, replaces T. Also, RNA has ribose sugar instead of deoxyribose sugar. The end of the gene is marked by a special sequence that signals the polymerase to stop. The RNA chain synthesized by the RNA polymerase is called a primary transcript. The sense strand is the same as the RNA being produced. The antisense strand is the template used to make the RNA product, so the RNA product is the reverse complement of the antisense strand. After eukaryotes transcribe an RNA, the primary RNA transcript is extensively modified before being exported to the cytoplasm. After all modifications, our product is called messenger RNA, or mRNA. The modifications include that a cap of 7-methylguanine, which is a series of an unusual bases, is added to the 5' end of the RNA. This cap is essential for binding the mRNA to the ribosome. A string of adenines, as many as 200 nucleotides, known as a poly-A tail, is added to the 3' end of the mRNA after transcription. The function of the poly-A tail is unknown, but thought to be linked to stability, translatability, and transferability of RNA. The poly-A tail can be used to capture mRNAs for study. When considering the gene sequence, the non-coding interruptions of the gene sequence are known as intervening sequences or introns, whereas the coding regions of the gene sequence are called exons. Although both introns and exons are transcribed when forming the primary transcript, these introns are excised or cut out to form the messenger RNA. This ideogram illustrates the primary transcript, which consists of introns, shown in red, and exons, shown in blue. As mentioned, one modification of the primary transcript is that the introns are excised or cut out, and the exons are spliced together end to end, forming the messenger RNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Note that only exonic regions will affect the amino acid composition of a protein produced by the messenger RNA. Alternative splicing is a process that allows a single gene to code for multiple proteins. In this process, particular exons of a gene may be included within or excluded from the final processed messenger RNA produced from that gene. This means the exons are joined in different combinations, leading to different or alternative mRNA strands. Consequently, the proteins translated from alternatively spliced mRNAs will contain differences in their amino acid sequence and, often, in their biological functions, as can be seen in this figure. We will not discuss alternative splicing, though tools to detect alternative splicing using RNA sequencing assay data have recently been developed. For now, we will operate under the usual paradigm that one gene makes one protein. In translation, the mRNA serves as the template for protein synthesis. The sequence of bases along the mRNA is thus converted into a string of amino acids. Each consecutive non-overlapping triplet of bases, called a codon, acts as the code to specify a particular amino acid. Interestingly, there are 64 possible codons, but only 20 amino acids. Therefore, there is room for redundancies. This provides a safeguard against small errors that might occur during transcription. Proteins fold to form a 3D structure. Thus, proteins are large, complex molecules made up of amino acids which do most of the work within the cells. Here is an ideogram illustrating the primary transcript being post-processed into mRNA, which moved into the cytoplasm, where then the ribosome helps direct the transfer RNAs, which bring the appropriate amino acid to each codon, or 3-base sequence, to grow the protein chain. This figure lists the 64 possible codons, or sequences of 3 RNA bases, and the corresponding amino acid each codes for. Notice if a base pair change results in a codon that codes for a different amino acid, this is known as a missense mutation. If a base pair change results in no change in amino acid, it is called a synonymous polymorphism, or silent mutation. Next, please watch the Transcription and Translation YouTube video.